Good afternoon. It's my pleasure to be here. I'd like to thank the organizers for running this event every year. It's personally my first time here, and I'm here to answer the question how the scientist can help people with rare diseases. I had to admit to myself that this was not easy to answer for me at all, but thankfully I was given the good advice of focusing on what I've done and how it could matter for people with rare diseases. I'm a molecular biologist, quite far removed from the clinic, from patients. I do basic research mainly. For my PhD degree, I researched the CF4 gene related to the Pitt-Hopkins syndrome. I moved into Germany where I researched development and evolution of the mammalian cerebellum. This is also relevant to rare disease. And as of now, a couple of months ago, I returned to my home in Tartu. I'm the principal investigator. Our topic is researching autism spectrum disorders in the developing cerebellum. Based on these three examples, I'd like to highlight a few points on how basic research can be important in the context of rare disease. Let's start with TCF4 and Pitt-Hopkins syndrome. I researched it in Tallinn in the working group of Professor Tõnis Musk. This is a regulator gene encoding regulator protein binding to DNA and affecting the expression of various genes in various tissues. We found that the structure of the TCF gene is a complex one. What I mean is, if you look at the scheme up there, each line signifies exons in the TCF4 gene. In order to create a functioning protein, first RNA is created, fragments are spliced, and sequences between them are then removed. These exons can be assembled in a number of ways, and for TCF4, the key mechanism is the use of initiator, the use of a variety of different initiator exons. Sometimes one is used, sometimes another one. For TCF4, there's 21 of possible initiator exons in total. As a result, it's possible to create different proteins of different lengths, this shorter and longer. In my PhD studies, research was released showing that mutations in one allele of the TCF4 gene cause Pitt-Hopkins syndrome to develop. This in turn leads to a moderate or severe intellectual disability, also spectrum disorders, and children born with this syndrome often do not walk, do not speak. When these works were published, Tõnis and I decided we are quite well familiar with the gene. We should find out how the mutations found in patients affect the functioning of this gene. There's a complex scheme displaying various mutations found in patients. There are deletions. Sometimes the entire gene or even a larger area has been deleted. Sometimes fragments have been deleted. And in patients, a number of point mutations have also been identified. But let's currently focus on the deletions. Some deletions, currently marked in red, especially in the early parts of the gene, along with the initiator exons, we found that these partial or shorter deletions do not remove all functioning of the allele. Alleles with deletions can oftentimes produce shorter fragment isoforms. The number of patients in our study was not large. We couldn't draw conclusive data, but it seemed that smaller deletions led to lighter expressions of disease. A few years later, a UK doctor got in touch with us. They had identified a patient with a lighter intellectual disability. For example, where patients were suffering of dyslexia, ADHD, but were still able to cope in life just fine. And those patients were found with partial deletions of the TCF4 gene towards the beginning of the gene. When we looked at the isoforms identified in the blood of those patients, which isoforms have been expressed, we found that the level of long isoforms was lower, and the general levels had not decreased. What we found was that years ago, when I went to a Pitt Hopkins conference in the States, I realized how important it is for patients to have a diagnosis, and secondly, to understand how different mutations affect the function of the gene. After I presented the work, I received very many emails from families as well as geneticists who asked about each gene, which isoforms remained and which didn't, or were present and which weren't. And naturally, we weren't able to give a prognosis for each particular patient due to a lack of sufficient information. But even small pieces of information on how the expressions can affect the life of the patient were important for them. The Pitt Hopkins Syndrome Foundation in the United States got in touch with us, with Tõnis Timmusk, to offer funding for the only time in my life where I didn't have to search for funding myself. They wrote to us, "Please keep researching, we will find the money for you." I have sometimes been asked why we research the Pitt Hopkins syndrome specifically. It's a rare disease and there were no known patients in Estonia at the time. And the response is that we didn't start out by researching Pitt Hopkins, but we started with a more basic question, researching the TCF4 gene. But by the time the disease was identified, we had done a lot of the work, we had a lot of knowledge about the dysfunctions, and this allowed us to proceed with researching the impact of the disorder. Moving on to the second example about the cerebellum. Many of us present today likely know that the cerebellum is important in keeping our balance in walking, but also emotional regulation, language, speech, cognition. Hendrik Kessmann's research group in Germany focused on the evolution of the cerebellum. We worked on we knew that the cerebellum has grown in the course of evolution, and we wanted to find out about the differences and similarities in different mammals' cerebellums. let's say, between a human and a mouse. The brain is vastly complex. It consists of various types of cells. Each cell type has its own function. They are interconnected. And in order to research such a complex tissue, it would be good to have information on each single cell type. These are historic depictions by Santiago Ramón y Cajal. He created drawings of single types of cells. These are the Purkinje cells in humans above and mice below. This was groundbreaking work over 100 years ago. But over a decade now, we've had new methods. A key method has been single-cell genomics. It allows us to receive information from hundreds and thousands of cells simultaneously. I believe we'll see lots of applications, including diagnostic applications for this technology. We used a single-cell sequencing method. We used tissues of different species, humans, mice, a few primates, and an opossum. And our biopsies covered various maturation stages of the organism. What we can do with this data, we can assess the relative density of various cell types in the tissue in different developmental stages. Here's the relative frequency of Purkinje cells in the cerebellum development. The human brain has more Purkinje cells than other mammals. We can compare gene expressions by the cell type. Here's the gene DSCAM connected to the Down syndrome as well as autism spectrum disorder. And we found the human astroglia cells here indicated in pink include this gene, whereas this type of cell in other mammals does not have this gene. And where the presence of gene differs between species, those genes are often implicated in diseases. This knowledge builds a foundation for us to choose modeling systems better in the future. We use mice frequently in creating models of diseases. However, it may not be the best model organism for all diseases, especially where we know there are interspecies differences. And this motivated the creation of the program I wanted to run when I decided to run when I came back to Tartu to research autism spectrum disorders and the development of the cerebellum. And I'm glad that a number of researchers and students have already joined my research group. We still have space. If interested, please get in touch. And I'd also like to say that there are hundreds of genes connected to autism. If we see the use of genes in the development of the cerebellum, we find that there is no single cell type where the expression would be higher than in others. The colors are different cell types, high expression on the right, low on the left. You see the variation is wide. And this is one of the questions we're trying to answer. In spite of the wide expression, are there any cell types that are more sensitive to changes leading to autism spectrum disorders than others? We also would like to know that in the hundreds of genes, of which we might be able to explore tens, whether the effects of the mutations of these genes have any similarities. And finally, whether specific aspects, whether human-specific cerebellum development aspects have a role in the development of the disorders. We used genetically modified mice as well as induced pluripotent stem cells discussed before to develop organoids. For me, the main question is, when comparing tens of mutations and tens of genes, whether we're able to locate similarities among different groups that might differ in turn from other genes and other mutations. For instance, if for one mutation-related disease a helpful drug has been found, then maybe it makes sense to attempt this treatment with other mutations that cause similar problems with the development of the brain. In summary, I'd like to say that basic research may not look this way to patients, but they're massively important in the long run for modeling diseases, understanding the mechanisms, and developing new treatments. We might not always know how a piece of basic research can prove useful. We are not able to predict everything, and sometimes solutions are found in unexpected places. And here, a long list of names of people who have been or are now my colleagues and who I'd like to express my gratitude to. This is a massively international group, and it is clear that we cannot do this alone. Research takes place everywhere. We need to collaborate to move on faster. Thank you. Thank you. Any questions from the audience? There's a question from the web. As a scientist, how much contact do you have with various patient groups or organizations? Not much, I'm afraid. My experience with patient organizations are mainly from the time of my PhD work, when I encountered Pitt Hopkins patient organization. I went to their conferences, I met patients and their families. This was an experience I'm grateful for. It was a motivating experience as a PhD student. Currently, I'm also part of a US network, so to say, the Siemens Fund, part of which is dedicated to autism spectrum research. There are large numbers of top researchers, and I've already found collaborators from this network. And yes, I can say networking is massively beneficial. Thank you, Maris Sepp.